Hi, everyone, and thank you for joining us today. Welcome to today's Microscopy Masters webinar titled Dissecting the Cellular and Molecular Mechanisms Regulating Vascular Function in Health and Disease by Professor Manuel Navedo. Before the presentation begins, we'd like to go over some quick house rules for the webinar. Firstly, the presentation will last about 45 to 50 minutes, leaving approximately 10 minutes for Q&A at the end. You are welcome to submit any questions you have throughout the webinar via the chat section, which is located in your panel. Furthermore, we will be answering your questions during the Q&A session. Within your panel, you'll also find a handout section. Here's a company brochure about O&I that we'd like to share with you today, so please feel free to have a look there. And lastly, please note that today's webinar is being recorded. It will be emailed to all attendees and also stored on the media section of our website for future reference. And now I would like to introduce you all to our speaker for today, Professor Manuel Navedo. Dr. Navedo completed his undergraduate and graduate studies at the University of Puerto Rico. He then moved to the University of Washington, Seattle to complete postdoctoral training in vascular biology and calcium imaging. And after several productive years, he joined the faculty of the Department of Pharmacology at the University of California, Davis, where he has been since 2012. And with that, I will now hand over to Dr. Navedo to begin his presentation.
Thank you so much. Thank you for the opportunity and to present about the work of the laboratory in this webinar series. By any means, I'm a microscopy master. I stand on the shoulders of visionaries and innovators that have developed microscopy tools and analytical tools that I have used to then dissect some of the molecular mechanisms regulating vascular function in health and disease, which is basically what I'm going to talk to you today. The model system that we use are vascular smooth muscle cells. These are cells that are in hollow organs and blood vessels. We can get them from both human and mouse models. This is more or less how they look. We can get them from the brain, the mesentery, the aorta. We do a cross section here. The smooth muscles are located in the tunica media of the blood vessels, just around here. This is where the blood comes through. And we can isolate these cells. They look more or less like these spindle-shaped cells that then we can, once isolated, we can use to record the activity of ion channels, look at signaling mechanisms. We can also use the isolated cells or the arteries to do mRNA, protein expression, phosphorylation. And my group have actually tried to use a multi-pronged approach to examine the cellular and molecular mechanisms regulating vascular function in health and disease. And we go from the biochemistry level, electrophysiology. We certainly use imaging approaches, and super resolution is one of those imaging approaches that is super hot at the moment. We combine that with in silico modeling, pressure myography, radiotelemetry, and laser spectral imaging. And the idea is to provide a broad understanding of how these mechanisms regulating vascular function are being regulated in health and disease. We go from the single molecule level all the way to the animal model. And we, in terms of disease, the lab is mainly interested in diseases such as diabetes and hypertension. Diabetes is a devastating disease that affects millions around the world. It has a major economic impact in the US and elsewhere, and it heavily impacts the vasculature. And I'll talk a little bit more about that in a few minutes. And hypertension or high blood pressure, it's estimated that actually half of the adult US population may suffer from hypertension, and therefore it will have an economic impact that is in the billions of dollars, not only in the US, but elsewhere. The primary is a primary risk factor for cardiovascular diseases such as heart attack, renal dysfunction, stroke, etc. And the thing that these two diseases have in common is vascular complications. Some of those vascular complications is altering the myogenic response. And the myogenic response is an intrinsic property of vascular smooth muscle is basically pressure-induced constriction. It was described by William Bayliss more than 100 years ago. And it is important because this myogenic response helps maintain constant tissue perfusion despite potential changes in blood pressure. This is more or less how it looks like. This is an artery, cartoon of an artery that was cannulated into glasses here just so that we can control pressure inside the cross-section of that artery. And here we're measuring pressure, diameter, membrane potential, and calcium. And when there's an increase in pressure, you can see that the diameter of the artery basically increases, but then it comes back down. That is myogenic response, pressure-induced constriction. And that pressure-induced constriction is mediated by a depolarization of the membrane potential of vascular smooth muscle cell that gets depolarized, and that depolarization increases the intracellular calcium of those cells, and that leads to contraction of the cells, as shown here. And through the work of many pioneer people, including Dr. Mark Nelson from the University of Vermont, we know more or less the mechanisms regulating this myogenic response. So an increasing intravascular pressure causes membrane depolarization that is caused by activation of TRP channels. That increases the open probability of this ion channel called L-type calcium channel, CAB 1.2. That will increase global intracellular calcium, and that causes contraction. And in this process, as you may expect, shown here, these L-type calcium channels play a very important role. Cell-type calcium channels are multimeric proteins, have basically 4 domains, 1 through 4, and each domain has 6 transmembrane domains, multiple accessory subunits. Cell-type calcium channels are expressed in excitable and not excitable cells. They regulate muscle contraction, cell excitability, gene expression, or excitation transcription coupling. And more recently, they have been found to be involved in proliferation and migration, cancer cell invasion. And as you may know, they are target of antihypertensive drugs. In vascular smooth muscle, these cell-type calcium channels are key. Calcium influx through these cell-type calcium channels is essential for vascular smooth muscle contraction. And as one might expect, they're regulated by many GPCRs. They can be regulated by GS PCR proteins, as well as GQ PCR proteins. And this is important because many of the disease states basically act through one of these GPCRs to actually regulate channel function. And that has been actually highlighted by, in part by my group and others. For example, we know that during diabetic hyperglycemia, increases in extracellular glucose levels as it occurred during diabetes, the L-type calcium channel activity goes up, and that is mediated by activation of a GS PCR receptor that actually stimulate the activity of the channel. Under conditions such as hypertension and angiotensin II signaling stimulation, which is enhanced during hypertension, we know that that could activate AT1 receptors that are GQ couple receptors, and that activation of that GQ couple receptor can also potentiate the channel. And let me just show you an example of how that looks like. This is actually a smooth muscle cell with ion channels, and this is a voltage, whole cell voltage clamp recording. In most of our experiments, we do this protocol here from -70 to +20 millimolar just to stimulate channel activity. And this is actually an example of a smooth muscle cell, freshly isolated smooth muscle cell. These are not culture cells, it's important. These are freshly isolated cells. This is the patch pipette. And when we stimulate with this pulse like this, under control conditions, and this is the same cell here, the black is the control trace, show this robust increase in the current with the membrane depolarization. But if the same cell is stimulated with a high extracellular glucose solution or hyperglycemic solution, then we see this potentiation of the calcium current. And this is quantified here. And likewise, if we take, here, the blue line is the control cell. And if we take that same cell and we stimulate it with angiotensin, we see this potentiation of the calcium current. And this is the amalgamated data here. And this is important because it showed that these pathological stimulus, such as hyperglycemia and enhanced angiotensin II signaling, can potentiate the calcium current. But unfortunately, these type of techniques doesn't allow us to visualize the spatial distribution of calcium entering events, where these events are occurring throughout the plasma membrane of the cell, and whether that correlate with actually this distribution of the ion channels. So several years back, 2005, I joined the laboratory of Dr. Fernando Santana in Seattle, and we started developing these optical patch clamping approaches in order to visualize the spatial distribution of calcium entry events. And for that, we use actually a technique called total internal reflection fluorescent microscopy, or TIRF, where an evanescent wave field is generated, and that allows us only to visualize calcium influx events that are very close to the cover slip. We put the cells there, we patch the cells to control membrane potential, and then we can visualize only what's happening at the plasma membrane of these cells. And this is more or less what we saw. And this is actually a hyperactive cell here. So this is the footprint of the cell. And all of those events there that you're seeing are calcium influx events. And one thing that we start noticing immediately is that this calcium influx event didn't occur throughout the entire plasma membrane of the cell, but occur in very specialized regions. We can take actually we can stimulate these cells with high glucose or angiotensin, and more or less this is how it looks. This is actually, again, the footprint of the cell. And these are two areas, regions of interest that we're plotting here. So this line here is the baseline, and the upward deflections that you're seeing are basically calcium entry events. And one thing that is apparent from this type of traces is that when we stimulate the same cell with high glucose concentration, 20 millimolar D-glucose, the activity of sites that were already active basically increases. And the activity of sites that were actually inactive before also increases. This is actually correlates with these increases in whole cell calcium currents that I showed you before. Let me show you another example here. This is cells under controlled conditions, just shown here. And if we stimulate with angiotensin, now you see more of these type of events. And we took one region of interest just as an example from these two cells here. And again, you're seeing here, the C is the closed state of the channel, the O is the open state, and we believe that this is the unitary quantal event shown here and here. And we'll notice that angiotensin actually increases that activity of the channel. So both high glucose and angiotensin alter the spatial temporal the temporal property of the channel, but also alters the spatial properties of the channels. And one thing that actually caught our eyes when we were actually looking at all of these traces is that we are not only increasing the open probability of the channel, but a lot of the events that you're seeing in this particular case here, or in this particular case here, are produced by two or more channels opening at the same time. And we have done a lot of experiments to corroborate that actually that is in fact the case. And as you may know, this is a process that is known as cooperative gating of ion channels, in this case of cooperative gating of CAB 1.2 channels. We can see that type of behavior using optical recording techniques or electrical recording techniques. And I've shown here, again, this line here is a baseline, and these upward deflections are the unitary events. And the dotted line reflects the single quantal event of opening of an eye of these L-type calcium channels. And this is done in murine arterial myocytes, as well as in TSA 201 cells expressing the CAB 1.2 channel. And even under controlled conditions, you can see some events where actually the event seems to be produced by two or more channels opening at the same time. In this particular example, there are at least 3 channels opening at the same time. And the cool thing about these trace is that after the closure of these 3 channels at the same time, you also have an opening of 1 channel there. And it's shown here, again, another example from TSA 201 cells overexpressing the calcium channel and the cooperative gating behavior. As I mentioned, we can also record these electrically. So this is not actually an artifact of the optical recording. You can see it in electrical recording. So this is actually a control trace here. This is a baseline. And these are the unitary openings of LCAB 1.2 channels. And in some examples here, we saw events that were produced by unitary events as shown here, and events that were produced what seems to be 6 channels opening at the same time, opening and closing at the same time, shown here. And why this CAB 1.2 cooperative gating is important? Well, it is important because it amplifies the amount of calcium that comes into the cell, and that could have a major impact in cell response. We have tried to address the mechanisms underlying this cooperative gating behavior. And more or less, we think that you have perhaps a certain number of channels around the plasma membrane. And under control condition without stimulation, many of them will be far apart enough that they don't interact with each other. But if you provide a stimulus, in this case, depolarization or say high glucose or angiotensin, they may likely come closer together. And that basically close association is driven by calcium calmodulin. And that close association is important because it then allows the channels to open and close in unison and to provide that cooperative gating behavior. For those of you interested in this process, we just published a paper last year, early last year, addressing that issue. But one of the key concepts of this cooperative gating behavior that one may wonder is that in order for to have channels that open and close in unison, they need to be close together. They need to be very, very close together. And the stimulus that is actually triggering this cooperative gating behavior have to actually likely have to stimulate that the channels get closer together to one another. So how do we address that issue? And that is actually was a hypothesis that we had when we start seeing this type of behavior. At the time, we didn't have the tools, but we now have. So how do we address the issue? And as you may wonder, we do that with super resolution imaging. We're using the STORM microscopy or stochastic optical reconstruction microscopy. And this is more or less how it works. Don't need to, likely I will not need to show you or describe this in major details, but in conventional fluorescent microscope, where all the fluorophore in the sample are fluorescent, their diffraction limited image overlap and they create this blur picture. However, with this STORM, it utilizes fluorophores or fluorescent probes that can switch between the fluorescent and the dark state so that in every snapshot, only a small optical resolvable fraction of the fluorophore is detected. And this enables the determining the position of each of these blinks with high precision from the center position of the fluorophore spot. And by doing that many times, we can then resolve a super resolution image. We can go from this blurred conventional image to this really nice tubular, microtubular, or really nice STORM image showing microtubules in a cell. We do have a ONI super resolution scope in the lab that we use to address the spatial distribution of CB 1.2 channels. And if you indulge me for a second here, I'll tell you a little bit about why I decided to go with the ONI super resolution system. First of all, it has a lateral resolution of 20 nanometer, so very good. This is actually our system. This is a microscope, as you can see here. This is the area where, or the casing where the lasers, the camera, etc., are located. And this is the computer. We put it in a stage so that basically we can use the area below for experiments. And we actually even added a nice monitor. The reason that I went with this system is the small footprint that you can see as you can see here. That it was important because space is always a limitation in laboratories. It's easy to use. Basically, we went from recording recording 6 or 7 cells a day to recording 20 cells a day on any really good day when the cells are really healthy and the system is and the conditions are working properly. So it speed up acquisition of images, etc. It's very simple to train new individuals to use the system. We've had an exceptional training and technical support. We got the system in the middle of the pandemic. And we were able actually to put it here by ourselves with the training via video conference. And that was relatively straightforward. And we had, and we still have amazing technical support from the system. As you can see here, the system actually is located in the middle of my lab, so it doesn't require a dark room. This helped, in my opinion, with training. So we can actually have somebody using the system, and if I know that they are doing something wrong, I can help them correct the imaging process relatively quickly. That also increases rigor and accountability of those collecting the images. It has nice integration with Cody, which is the analysis software that is created by ONI. And it's very versatile. It has third system, high-low, confocal options, and temperature control. Here, these are just a few examples. As I said, this system is actually standing in the middle of the lab. This is Dr. Jung Hong, a lab postdoc in the lab, actually collecting images just a few days ago. And actually, this is Pia. Pia is a high school student. So we're training high school students to use this system. And Pia is very good already at acquiring images and at analyzing those images. So these are some of the examples of how it looks like. This is actually the software. Here we use actually a fluorescent bead, and we move that fluorescent bead every 20 nanometers, and then we basically do a line profile, and you can see here that indeed it's actually it's able to detect every 20 nanometers changes. These are actually like the GATA other specs with three dots that are supposed to be 40 nanometers apart, and indeed that's what we are recording. And when we were basically starting with the system, we wanted to make sure that we were actually basically imaging what we needed to do, what we needed to image. So we got actually a sample from ONI. These are shot cells, and we're staying with microtubules. And in just a matter of days, we were actually able to image this nice microtubular staining. Eventually, we went on and we took cardiomyocytes. We stained it to look at the CAB 1.2 channel. And here you can see actually clusters of CAB 1.2 channels that are actually very much in this titubular region pattern that is expected for this channel. And some collaborators are now starting to look at neurons. And this is a staining of PSD95 that was analyzed with Cody. And this is in neurons, and this is images were provided by Chen Yang Ho and Johannes Hell here at UC Davis. So in terms of the vascular imaging of the CAB 1.2 channels, when I'm talking about clusters, it's two or more channels or like a number of channels in the area. This is actually the blinking of how it looks. And then here I'm just showing you the C projection, an average C projection of that movie. This is actually the rendering of the image. I apologize because the image looks a little bit darker than what I would like it to see. But these are some region of interest showing the CAB 1.2 clusters in these cells. And one of the things that we wanted to do with this technology was to figure out whether high glucose or angiotensin II signaling or diabetes and hypertension will have an impact on CAB 1.2 clusters in vascular smooth muscle. So we start doing that. Here, this is a wild-type smooth muscle cell based in regular 10 millimolar glucose conditions. We saw like these nice clusters of CAB 1.2 channels. But unexpectedly, when we when we treated the cells with high glucose, what we start to notice is that the clusters of the CAB 1.2 channels became bigger, and that data is actually quantified here. Okay. Likewise, when we did the experiments under control conditions, again here this is the control. when we treated wild-type cells with angiotensin, we start to notice that the clusters of these CFB 1.2 channels became bigger compared to the control conditions. So these results suggested to us that high glucose and angiotensin increase CFB 1.2 clustering in vascular smooth muscle cells. And it's a behavior that actually wasn't actually described previously, but that it could underline this increase in cooperative gating that we saw using electrophysiological and optical recording techniques. So subsequently, we knew that diabetic hyperglycemia proceeds through this pathway, the GS PCR pathway, and that angiotensin and hypertension could proceed through this GQ pathway. So the question that we had was, how are these two pathways then stimulating the activity of these L-type calcium channels? And as it turns out, there is a serine, a position called serine 1928 that is known to be a readily phosphorylated pKa site. And that potentially could be phosphorylated by this site. And in other studies from Stephen Mark's group and others, we also knew that this same position could be phosphorylated by PKC, which is activated during the GQ-PCR activation. So this was very intriguing because it potentially suggests that this site could be a master regulator of the channel. And we certainly were intrigued trying to figure out whether indeed this site was important for regulation of the channel by these two signaling pathways. So we're fortunate we have a mouse in which serine 1928 is mutated to an alanine, and that prevents the phosphorylation of that site. And here, showing you some examples. This is the data that I showed you before, where high glucose increased clustering of CFB 1.2 channels when compared to control conditions in wild-type cells. However, when we did the same treatment options in knocking cells, shown here, now what happened is that preventing the phosphorylation of 1928, also preventing the high glucose induced increased clustering of CFB 1.2 channels. And that is actually high quantified in this case here. And likewise, the angiotensin induced increase in CFB 1.2 channel clustering in wild-type cells, shown here, was completely prevented in knocking cells, highlighted here. So very intriguing, very important findings suggest that the phosphorylation itself seems to be triggering the clustering of the channels. Now, these are super resolution techniques, and we wanted to actually go a step further and validate these super resolution results. And in order to do that, what we ended up doing is using proximity ligation assay with monoclonal antibody for the CIB 1.2 that we tag with either the plus probe or the minus probe. And if the channels were coming together, we were expecting to see more of these PLA pontas. If they were not coming together, then we will see less PLA pontas. And consistent with the super resolution data, what we find is that in wild-type cells treated with angiotensin, there's an increase in PLA pontas that correlate with like the super res data shown here. If we block a PKC, then we prevent the angiotensin increase in PLA pontas, suggesting that in this particular case, angiotensin is proceeding through PKC to increase the clustering of the channels and the number of PLA pontas. And if we prevent the phosphorylation of the serine 1928 site, we prevent also the increase in PLA pontas, like similar to the results that we observe in super resolution data. So these two techniques together provide really strong evidence that serine 1928 phosphorylation is important to increase this CAB 1.2 clustering in response to high glucose or angiotensin 2 signaling. We can see actually, so we have access to samples, blood vessels from humans, non-diabetic in human patients. And actually, to correlate all of this information that I show you, this is the blood vessels that we use, very small blood vessels. When we did Western blot analysis, we found that the serine 1928 phosphorylation site is significant. The phosphorylation of this site is increased in diabetic arterial lysates compared to non-diabetic arterial lysate. And that actually was important because if, as indicated before, the phosphorylation of this serine 1928 site is very important for increased clustering. And look at what happened in some muscle cells from non-diabetic and diabetic patients. The non-diabetic patient, the CB 1.2 clustering of the channels is about the same as in control conditions in animal models. But in diabetic cells, we saw this increase in CAB 1.2 clustering, which actually correlate then again with this increase in serine 1928 phosphorylation, suggesting again the importance of this site in increasing CAB 1.2 clustering in diabetic vascular smooth muscle cells. We have gone on and we wanted to actually test a little bit more whether the 1928 site was actually important. And for that, we created animal models of diabetes and hypertension. In the wild-type diabetic models, we see this massive increase in CAB 1.2 clustering compared to sham conditions, but that is prevented in knocking diabetic cells compared to the controls. Likewise, in wild-type hypertensive mice, there's this massive increase in CAB 1.2 clustering compared to sham conditions, and that is actually prevented. If anything, the clusters and at least the cluster density goes a little bit further down in the knocking hypertensive cells. We validated the super resolution data with proximity ligation assay in both diabetics and hypertensive animals. You can see this increase in PLA pontas in the wild-type diabetics, but not in the knocking diabetic cells. And again, an increase in PLA pontas in the wild-type hypertensive cells, but not in the knocking hypertensive cells. So this super resolution have revealed that CAB 1.2 spatial remodeling is mediated by serine 1928 phosphorylation in both diabetic and hypertensive vascular smooth muscle cells. One thing that I want to correlate in the next couple of slides is that this actually increased clustering that we're seeing here is actually functionally and physiologically relevant. And we tested that, we went back and we tested that the functional and physiological relevance. We started by looking at actually electrophysiological recordings in the single in the cell attached configuration of the channels. And what we are finding is that in wild-type hypertensive cells, there's an increase in the activity of the channels, but also that an increase in this cooperative gating behavior, where many of the events that you're seeing here are produced by two or more channels opening at the same time. And this actually increase in activity and cooperative gating behavior in wild-type hypertensive cells is actually completely prevented in hypertensive knocking cells. We observe similar results in diabetic vascular smooth muscle cells, and I don't want to bore you showing additional data about that. But these experiments are very important because they provide a link between the CAB 1.2 spatial redistribution and the temporal changes in this cooperative gating behavior that we're seeing in vascular smooth muscle cells during pathological conditions. At the physiological level, we know that actually if we do we measure myogenic response using pressure myography in wild-type hypertensive animals, we see this increase in myogenic response in wild-type hypertensive arteries compared to normotensive sham conditions, but that changes in myogenic response completely prevented. We have gone a step further. We have measured blood pressure in animals using radiotelemetry. And what we're seeing is that the wild-type hypertensive have this massive increase in mean arterial pressure compared to shams. And even though the knock-in hypertensive animal have a significant increase in mean arterial pressure compared to the corresponding sham, that increase is significantly smaller when we compare it to the wild-type hypertensive sham, suggesting that this serotonin 1928 phosphorylation site is not only important for basically the clustering and comparative gating behavior of the channel, but it's also important for enhancing tone and alterations in blood pressure regulation during hypertension and diabetes and other conditions. So I hope that the data that I have shown you convince you that the use of all of these techniques allow us then to correlate changes at the single molecule level of ion channels to what could happen all the way to blood pressure measurements. So in summary, I think like what I want to highlight again is that we have leveraged this super resolution imaging and other techniques to gain insight into mechanism regulating ion channel, spatial, and temporal properties, and how those changes basically translate to pathophysiological responses. So we know that during diabetic hyperglycemia or hypertension and angiotensin II signaling, these two processes basically act on this serine 1928 site on the CAV1.2 channel to increase phosphorylation. That increase in phosphorylation increases the clustering of the channels. And with that increasing clustering of the channels, you have actually more channels that come closer together and can now act in unison to open and close and in a cooperative manner. That will result in calcium amplification. And with more calcium coming in, we know that that could cause contraction. It could also change gene expression, and that's something that we're looking at the moment. But with the discovery that all of these pathways proceed through this serine 1928 phosphorylation site, it also opens the opportunities for therapeutic intervention. So one could imagine a small molecule that could actually prevent or reduce the increased phosphorylation of this site during these pathological conditions in order to correct the aberrant CAV1.2 channel function, rather than blocking the channel, which is basically what current antihypertensive medications do. So with that, I'll stop here. I know I'm a little bit earlier, but I want to acknowledge the people that did the job. Madeline Nieves Cintron is my partner in crime and being a major influence in the work. The work was done by Victor Flores Tames, Jun Hong, and Miguel Martin Aragon. And this is the team that we have at the moment. I'm very proud of this diverse group of individuals that are doing their best to further and to use these technologies to further understanding of vascular function in health and disease. I want to actually recognize Dr. Fernando Santana because he was the first one who actually came up with the idea. He said, okay, so there's this new technology available at the moment. Can we leverage to use it and to learn more about that? And he has been a visionary and in many different areas of cardiovascular research. And we do what we do these days in part because of that vision that he has. Collaborators here at UC Davis and elsewhere that have provided tools, material, etc., and the funding agencies. And with that, I'll stop. I'll thank all of you in the audience. And yes, I finish here with thank you so much and gracias. And if there's any questions, I'm happy to address them. Yes.
Wonderful. Thank you so much, Dr. Navedo. That was really wonderful. And we're now going to open up the floor to questions, which Dr. Navedo will be answering live. Once again, if you have any questions, please just feel free to drop them in the chat panel, and we will gladly be answering them now. And it looks like we already have a good amount of questions in for you. So we'll start with the first one. How does serine 1928 phosphorylation promote CAV 1.2 clustering?
Yes, so thank you. That's a good question. The hypothesis that we have at the moment is that the phosphorylation will lead to an initial increase in the activity of a single channel. That actually will increase intracellular calcium, and that increasing intracellular calcium will be essential to trigger the signals that will promote either more trafficking of channels from the inside of the cells to the plasma membrane, or lateral diffusion of channels that will then increase that clustering behavior. That's our hypothesis at the moment. We have some evidence that if we treat the cells without, like when we treating the cells with like angiotensin or high glucose, but we don't have calcium in the bath, the process doesn't occur, which actually support that hypothesis. But we still have a lot more work to do, and yes, and hopefully in the next couple of months or so, we'll have more details about that specific mechanism.
Wonderful. Next question is, can you say something about how the samples were prepared for the nanoimaging?
Yes, as you may know, I might be the wrong person to answer that question because unfortunately, I'm not that much in the lab at the moment. But we do, so a couple of things that are important here. We do see, I think it's we use different type of buffers. I will encourage you all to go to our circulation research paper from 2022, where we actually have a very detailed description of the different fixation process and the blocking process, because they are extremely, extremely important. One thing that I will say, though, that is that has actually worked well for us is that when we start before the Nanoimager, we were creating our own buffers in the lab, and they were working just okay. They were giving us the samples, but we start actually using the ONI buffers for super resolution, because the quality of the blinking that we're getting is substantially better than when we do our buffers in the lab. And that has been a major, major point of improvement in how we do these experiments.
Wonderful. Next question is, do you consider changing the serine for threonine or tyrosine, an amino acid that can possibly be phosphorylated too?
Yes, that's a good question. We definitely could do that and do it in an heterologous expression system. And likely there is some evidence along those lines from Valentina Di Biasi in Austria, where she did that type of analysis, and using immunoGOL technology, she was able to see that when she mutated the serine 1928 amino acid, she could actually increase the clustering of neuronal CAV 1.2 channels. And in our case, we are it's something that is interesting to us, but we wanted to keep it, we wanted to use actually native, freshly isolated native tissue, because that way we can correlate it more to what will happen during pathological conditions and human pathology. So, but yes, I mean, like the expectation would be that if that phosphorylation site is replaced with another amino acid that either like mimic phosphorylation or that is still causes phosphorylation, that likely the clustering of the CV 1.2 channel will increase.
Great. Next question. Do the GQ and GS coupled receptors target the same CAV 1.2 channels?
Yes, that's a really good question. At the moment, I don't know. But if I have to speculate, I will say no. And the way that I will think about this is that perhaps you have different populations of calcium channels, CAV 1.2 channels that are targeted by different GPCRs, and that they could actually then trigger activation of the channels under different conditions. That is something that we are exploring at the moment. The issue with the AT1 receptor, and if I show you here, the issue with trying to address that idea is that there's not that great antibodies for AT1 receptors, and therefore the results may not be definitive at that time. But definitely is something that that would be interesting and to us, or that is interesting to us, and that the expectation is that yeah, the different GPCR will target different pools of CAV 1.2 channels.
Okay. Next question. How long does it take you to acquire one D-storm image to obtain significant information from your samples?
Yes, thank you for that question. I mean, the way that we're doing the recordings, we are doing 10 millisecond exposure time at the moment. I mean, with good antibodies, and this is very critical. With good, well-validated antibodies, we're doing 10 millisecond exposure, and we're collecting about 30,000 images, and that is about between like a closer to 10 minutes or so. So it helps us acquire sufficient amount of images the day. One of the advantages of the ONI system that we have, and the reason that we are able to acquire it fast, is that we're able actually to track the entire field of view or the entire cover slip, and kind of identify where the cells are in those cover slips. So when we do that, then because we have that map of where the good cells are located, we can actually acquire one image, finish with that image in 10 minutes, 15 minutes or so, and then go quickly to the next cell that is there and acquire again another cell. So it's an advantage of the system, yes.
Great, on to the next question. How do GQ and GS coupled receptors stimulate CAV 1.2 activity?
Yes, thank you for the question. I was actually expecting it. So like this is more, this is how we think this is happening. And the reason that why I think actually that the different GPCRs could be targeting different channels. So for in the case of the GS couple receptors, we know that there are all of these proteins that are basically in complex with the calcium channel, and that they need to come closer together, held by this scaffold protein ACAP5, and which basically hold the channels in place, and the different components in place to regulate the channel. And in the case of the GQ, it's again, ACAP5 is important, but we think that it goes through PKC, and PKC then can phosphorylate the channel and increase activity. So one of the things that that we found early on, actually, with super resolution microscopy is perhaps some in the audience may not know this, but cyclic AMP PKA regulation in vascular submuscle is typically associated with relaxation. And during this, in response to elevations in glucose, we know that this pathway gets activated and induces contraction. So this was a dichotomy in the field. And the way that we use super resolution to address that is we start looking at the localization of calcium channels with PKA and with other components of these pathways. And what we know is that like these different components of these pathways, or that the calcium channel is associated with subpopulation of components of these pathways. So it's not with all of them at the same time. So that's how we actually are able to reconcile the cyclic AMP PKA inducing construction rather than relaxation in this muscle cell. And it's an idea that came out of basically doing super resolution and trying to figure out where the different proteins are associated with each other.
Okay, on to the next question. Do all CAV 1.2 channels need to be phosphorylated to undergo cooperative gating?
Yeah, it's a really good question too. Not necessarily. You could have, for example, one calcium channel that is phosphorylated, and if that and if that channel actually basically have an increase or allow an increasing intracellular calcium to trigger actually the clustering of the channels, then channels like such as this one that is not phosphorylated but is really close to the phosphorylated channel, we know that that phosphorylated channel, hyperactive channel, could then hijack the activity of the unphosphorylated channel and make it behave as the phosphorylated channel. So you don't need to have all the channels within the cluster phosphorylated in order to trigger this cooperative gating behavior. And that is consistent actually with these transient effects of the cooperative gating behavior that we're seeing there, that it's not an all or none thing, but it's actually very graded at the time.
Okay. Next question. How many channels can you use for imaging? And have you tried to look for 3D changes?
Good question. Right now, we are doing 2 channels. We are doing 647 because it's the best blinker. And we landed on using the 561 for the second channel. With the ONI, we are limited to 2 channels because of the way that the system is developed. So we are only doing 2 channels at the moment. And then the second question was, I'm sorry, I forgot the second question.
And have you tried to look for 3D changes?
Yes, we haven't done the 3D changes, but that is a possibility, especially with the ONI Nanoimager, that you could do that, but we haven't done it. Yes, and something that we like to do. It's just it hasn't reached the priority list at the moment.
Okay. Next question. What fluorophores did you use to label your proteins of interest for D-storm clustering imaging? And did you encounter any challenges during labeling?
Yes, so right now we are using the Alexa 647 for the labeling. For the calcium channel itself, we're using a custom antibody from one of our collaborators, which it's perhaps one of the best for these type of proteins. For the other proteins, we need to do validations. We need to make sure that the antibodies are good and that the blinking is good. For the 647, we are using a combination. We can use Alexa 561 or the Atto 561. Those are the 2 ones that we are trying the most. But again, it's a trial and error type of thing. And in terms of labeling, yes, we're encountering issues at the moment, just depending on the protein that you're looking at, the quality of the antibodies. One of the proteins that we're looking at right now doesn't have that many good antibodies. And unfortunately, if we need to do 2-color imaging, then we're limited by the species in which the antibody was grown. And etc. So we need to actually you have to do really good validation methods. And one of the things that we're trying to do now is basically, like trying to find conditions that will give us or that will allow us to basically image clusters of proteins of interest that we think may be relevant.
Great. Okay, we have a few more questions left, and we're getting close to time. The next question is, are there any studies showing a correlation between mutations in serine 1928 with disease prevalence?
Not that I know at the moment, but there are mutations in the CV1.2 channels that causes the Timothy syndrome mutations, so 406 to arginine mutation. And those mutations, I think these are again, human mutations, naturally occurring human mutations. And they do, we haven't done the experiments, but if I were to speculate again, if you allow me to do that, my expectation is that those mutations will increase the clustering of the channels. And the reason that we know that is because we have done some experiments where channels carrying that Timothy syndrome mutation do have more cooperative gating behavior. And if that is the case and the clustering, increased clustering underlying that cooperative gating behavior, I would expect that these Timothy syndrome mutations will indeed trigger more clustering of the calcium channels.
Thank you for that answer. Next question is, what is the common buffer composition that works for both of the dyes?
As I mentioned, I mean, like we try to use our own buffers, but quickly realized that the ONI buffer was superior for basically getting high quality links. So that is a proprietary composition, and I will encourage the audience to reach out to the ONI team to learn more about that and whether you can get some samples maybe or something.
Okay, next question. And we are very close to time, so I think this is the last one that we are going to take. Can you elaborate more about the staining protocol in DStorm? Are these live or fixed cells? And what is your experience with live imaging?
These are fixed cells. We fix them. I'm blanking on the fixation reagent that we use right now. I apologize for that. But these are fixed cells. And the way that we do the experiments is before fixing them, we treat them with either high glucose, angiotensin. And if the cells come from diabetic or hypertensive animals, then that's it. We fix them quickly. But typically, you treat the cells, freshly isolated cells, you treat them with whatever you want, and then we fix them, and then we use them for the imaging. In terms of live cell imaging, one of the things that we're trying to do at the moment is actually, rather than using fixations or antibodies, we're trying to actually look at the calcium channel in its native environment, native calcium channel in the native environment, and see whether we can actually detect live this clustering of channels. And for that, we have developed some tools that hopefully in the next couple of months or several months, we're gonna be able to share with the community. But I don't have that much experience looking at super resolution imaging in live cell. That's something that I don't have that much experience at the moment.
Okay, and with that, it seems that we are about out of time, so I do need to conclude the webinar. If we were unable to answer your questions, we will ensure to send them to Dr. Navedo on your behalf. And once again, thank you for the presentation, and thank you to all the attendees for joining us today. Upon exiting the webinar, you will receive a survey. Please fill out the survey to provide us feedback on your experience today. And if you are in need of being contacted directly by ONI for more information on our offerings. And thank you again, and we hope that everyone has a great rest of the day.
Thank you.